Thank you very, very much, Thomas Martin Urban, for those very nice words. And thank you to the Sjöberg Foundation for this honor. It is a true highlight of my scientific career. I really, really appreciate this very, very much. And the prize selection committee, of course. And so I would like to tell you about the work in our lab and the discoveries to target K-Ras. And so first I will explain that I do have some financial conflicts of interest with companies that have taken the drugs from our lab and worked on them to get them to the point where they could enter the clinic, and those are shown here. So cancer is a genetic disease of cells, and single cells acquire mutations. And these mutations occur in proteins, one family of which we call oncogenes. And these are the gas pedal, if you will, of cancer, the instructions to grow. And mutations in K-Ras occur at one amino acid, most commonly the glycine 12 position. And when this mutation occurs, then the cell becomes independent of external growth control, and it acquires its own ability to decide when to divide and grow, and to prevent itself from dying. And that creates a signal that goes throughout the cell, but the core of it is really through a protein kinase called BRAF that sends that signal to grow. So this is a kind of disease of signaling or instructions for the cell. Now, luckily, when cells acquire randomly this point mutation, that does not give them the ability to form a full tumor, because there are strong tumor suppressive mechanisms in the cell that Martin mentioned. And these, one of the core tumor suppressive functions are controlled by a protein called P53, which is a transcription factor that binds to DNA and senses the oncogene-induced stress given by the K-Ras mutation. And when P53 is activated, it sends a stop signal, orchestrated signal to stop the cell from dividing. And as long as P53 is active, that stop signal is controlling the cell growth. But that cell is also subject to mutational processes, and P53 itself is mutated in many, many tumors, and then that takes the brakes off of the growth, and now the cell has unlimited proliferative potential. And Thomas told you that this was one of the most frequently mutated genes in cancer, and to give you an idea, every year in the world, there are approximately 18 million cancer patients. And about 2.5 million, 3 million patients will acquire a K-Ras mutation, and about 9 million will acquire a P53 mutation. So these are the two most frequent mutations on the accelerator and the brakes of cancer. Now, when the cell is dividing and it's going through the cell cycle, acquiring new mutations at a very rapid rate, because it can't quite orchestrate the normal DNA error correction mechanisms, then the cell picks up other mutations. And these you can think of as private mutations, or very rare mutations in genes that themselves may not give the instructions to grow, but they acquire these mutations. Now, when a cell contains 100, 1,000, 10,000 mutations, it starts to look like non-self. So it starts to look different from all the rest of the cells in the body. That gives a chance for the immune system to find that cell. The way the immune system finds these cells, one way, is by processing all the proteins into peptides. And you can see anytime there's a dot, that's one of the mutations in the proteins. And then it becomes processed through what's called the antigen processing pathway, presentation pathway, and it appears on the surface of the cell on class 1 MHC, one of the most important components of our immune system. And if that peptide contains the mutation and a T cell is produced by the body to see that mutant, what we call a neoantigen. It's a new antigen that's present because of the tumor. Then the T cell can kill the tumor. Now, that is a many-stepped process. Many things have to go right for the immune system to see the tumor. And what's worse is the tumor can fight back against the immune system. It does that by expressing proteins like PD-L1 and PD-1, which are so-called checkpoint engaged molecules that turn off the T cell and doesn't allow the T cell to kill the tumor. And now we have revolutionary checkpoint therapies developed by the first Sjöberg Prize Award recipient, Jim Allison, that now can inhibit those checkpoints and reduce the negative signal and allow the T cell to kill the tumor. So these are three of the hallmarks. There are more hallmarks of cancer, but these are the three that are, we think, are the ways that we can re-engage and develop drugs. And so most drugs block the oncogenes. The first one of these was the Gleevec, the BCR-ABL inhibitor, by another Sjöberg Prize Award winner, Brian Drucker. We don't have so many ways to re-engage tumor suppressors. That's something that we think will be coming in the coming years, and that has a very powerful effect on tumors, but it's much harder to understand how to resuscitate a protein rather than to inhibit a sort of active protein. And then, of course, with the checkpoint therapies and cell therapies, there's lots of advances in immune oncology, and I'll tell you about one of those that we've developed at the end. So what gave rise to the idea that this first oncogene discovered, the most common oncogene to be discovered, what gave rise to the fact that this was an undruggable protein? And how do we block its function? As Thomas said, we discovered it in the early 80s, and only now until 2021, and the first drug was approved, the second drug was approved last year. Why did it take so long? And you could say, well, it just takes a long time to develop drugs. But the very next gene in the pathway is also an oncogene. It's actually a very common protein kinase oncogene, BRAF, and it was discovered in 2002. First time it was identified as a human oncogene. The drug that treats that disease was approved 9 years later. So in that case, it only took 9 years to discover the drug. Why did it take almost 40 years in the first case? Well, as Thomas said, it is really a property of the biochemistry of these two nucleotide binding proteins. Kinases have a weak affinity for ATP, 20 micromolar, whereas GTPases, in particular K-Ras is an outlier here, it binds and holds on to GTP with 20 picomolar. That is a million-fold difference in grabbing onto the nucleotide. And so it is really impossible, probably, to ever discover a drug that will block K-Ras function in an analogous way to the many drugs we have that block kinase function. So actually, the first strategy developed to try to treat K-Ras-driven tumors was to block its localization to the membrane. And this, I'll take you through that story because in the 80s and 90s, this was a very, very promising strategy, but I'll explain that there were some unexpected biological twists that sort of made that challenging. So, this is just one of the many timelines one could make for K-Ras, but the first RAS allele gene was discovered by Jennifer Harvey in 1964, and then the Kirsten or K-Ras gene was discovered by Werner Kirsten several years later, and then the human oncogene in the early 80s was discovered by multiple labs working very hard on sequencing and tumor virology, and then human sequencing. Now, once these were discovered, people tried to uncover the biochemical mechanism of its function. It was the first of its kind, so this all had to be discovered from the beginning. And One of the, I think, important discoveries was in 1984 that lipidation of K-Ras was important. A lipid was attached to K-Ras that allowed it to go to the membrane. And this mutation, lipidation is attached to a cysteine on the protein. And if you make a cysteine to serine mutation, the lipidation cannot happen, and therefore it doesn't go to the membrane, and it's completely inactivated. So this gave off a fantastic hypothesis that if lipidation could be prevented, then RAS-driven tumors could be treated. And actually, Brown and Goldstein, who also made foundational discoveries in the cholesterol pathway, noticed that they identified the protein that added the lipid group, which was farnesyltransferase. And then they collaborated with scientists at Genentech to make inhibitors of the farnesyltransferase. And all of this work proceeded very logically, very beautifully from biochemistry, enzyme isolation, drug discovery. And what is shown here is how RAS is made after it is translated by the ribosome. You see this is shown for HRAS. The color domain shows the structured GTP binding domain, but each of the RAS isoforms have a tail, a flexible tail. They include a cysteine, and that's where farnesyltransferase, or FTAs, adds farnesyl pyrophosphate onto the cysteine. And FTIs, or farnesyltransferase inhibitors, were readily discovered, and so really powerful compounds that could block the function were made. And when RAS first gets farnesylated, there are two more arrows there that are here and here. And actually, Martin made many important discoveries in understanding these enzymes. They process the protein, having a protease cleavage, and then at the end add a methyl group to make a methoxy, a methyl ester. So there are multiple steps in the processing of this pathway. But importantly for this talk, farnesyltransferase inhibitors blocked RAS localization and treated RAS mutant tumors. So this strategy looked more and more attractive because it was very logical. Now I need to tell you about the differences between HRAS and K-Ras. They are several important differences. They each have a very conserved GTP binding domain, but you'll notice their C-terminal tails are different, and their frequency of inducing cancer is dramatically different. HRAS is found in bladder cancers and several others, head and neck cancer, but K-Ras is mutated, as I said, the most common oncogene, so it's very frequently mutated. Now, interestingly, the enzymes that recognize to add the farnesyl group are recognizing the linear sequence of this flexible tail, and the primary sequence is the so-called CAX box, and that is the cysteine, aliphatics, and then various residues. But there was also another sequence upstream that was later found to be also important, and that is this sequence here, which has various different amino acids upstream of the CAX box. Now, this was all being discovered and identified, and people thought, well, if we can block HRAS localization, K-Ras localization will also be blocked. Now, K-Ras has another variant that it has alternative splicing forms that can have one form, the 4A form, that is kind of similar to the HRAS tail, and then one form that is very foreign, very different than the others. And you can see it has polylysines, and as you know, polylysines are positively charged, and positively charged tails like to associate with the negatively charged inner leaflet of the membrane. So K-Ras4B has an extra way to find its way to the membrane. So it's an outlier. And then subsequent studies told us that of both of these two forms, the most important form for most cancer signaling is the 4B form, which is the most unusual tail. So how does that play out when we look at farnesyltransferase inhibitors in K-Ras mutant tumors? And here, the drugs still blocked the enzyme very well, and then they produced the cysteine that was free. And unfortunately, the cell had a bypass mechanism, a different enzyme called geranyl transferase that also could recognize this tail sequence and add one extra unit, an extra isoprene unit, onto the tail, and then this still allowed processing by the subsequent enzymes and membrane localization. So because of the unusual part of the 4B isoform and K-Ras being the more frequent oncogene, all of the farnesyl transferase inhibitors put into the clinic were given to K-Ras mutant patients. And they were subject to this biological bypass mechanism, and therefore they didn't receive clinical benefit. And once this was really clarified in biochemical studies, again, by there are many labs, but including Brown and Goldstein, it became clear that the farnesyl transferase inhibitors alone would not be the approach that would allow people to treat K-Ras tumors. So this, besides the affinity for the nucleotide and this farnesyl transferase complexities, and we know there's much more complexity as well in the downstream processing enzymes. Martin's lab has really shown very surprising results of trying to make drugs or genetic models of other ways of blocking membrane localization, and there's a whole world of biology there still to uncover. So let me now explain the biochemical mechanism of the glycine 12 mutation. Why do mutations focus at that amino acid? And to do that, I need to tell you about how RAS is a signaling protein. So RAS is shown here, and it has two nucleotides that it binds to, either GDP or GTP, the diphosphate or the triphosphate. And in either of these two nucleotide states, we know that there are so-called switch regions or loops of protein of amino acids that are differentially stabilized in the GTP state or the GDP state. In the GTP state, the surface of the protein is complementary to downstream effectors like PI3 kinase, which I haven't told you about, but I told you about BRAF, and these send positive growth signals for growth and for mitogenic signaling. So the important feature of this protein is it cycles, and the way it cycles is that it is controlled by other proteins in the cell. In order to go from the off state to the on state, is used a guanine exchange factor, a protein that pries that high affinity nucleotide off and then lets the new GTP load onto the protein and signal. But now the part that the cancer cell cares about is how it can be turned off. And there are two ways for this to be turned off. In the fast way, it uses another protein called a GTPase activating protein, or a so-called GAP protein. And then in the slower path, it uses intrinsic hydrolysis, just the protein itself. So now let's zoom into this interface between RAS and GAP. And there is where we find the glycine 12 position. It is exactly at the interface between RAS and the GAP protein. And the GAP produces an interface that importantly includes this arginine that adds positive charge to the negative charge environment of the phosphates, so then it can hydrolyze very quickly. The glycine 12 side chain is pointed right at the arginine, the catalytically important residue, complemented by the GAP protein. And since glycine is the smallest amino acid, any other amino acid is bigger and pushes the arginine away. And that is the beginning of the kind of story of our discovery. If we actually look across different tumors and we ask, what does that glycine become in different tumor types, you can see that these are the 12 most lethal cancers in the United States, data from 2014. In green is the incidence of each cancer. In yellow is the mortality of that cancer type in that year. And in blue is the frequency of patients each year that will have a K-Ras mutant in that tumor. So you see that the K-Ras tumors are found in lung, colon, and pancreas. And in pancreas, you can see it's 80% or more of pancreatic cancer patients have that K-Ras mutation. Now, what's interesting is that each of the different histotypes have a different balance of mutations of what that glycine turns into. In pancreatic cancer, it's aspartate, valine, cysteine. The same order, but different distribution in colon. And very interestingly, cysteine becomes the dominant mutation at the glycine 12 position in lung cancer. And one reason for that is that the carcinogens in cigarette smoke produce the kind of DNA change that give rise to the codon for cysteine. And then if we sum all of these up, you can see that this is the sort of distribution across the 25% of cancer patients that have K-Ras mutants. So many people began really to think about how to drug the aspartate, because it's the most common amino acid that K-Ras becomes, and especially in pancreatic cancer. However, we looked at this distribution pie chart in a different way. We said, which of these amino acids give us a chemical opportunity to make a compound against this very difficult to drug protein? And cysteine is unique among the 20 natural amino acids in being the most nucleophilic, or in other words, has the easiest chance to make a covalent bond with a small molecule drug. And so with that realization, we focused not on the most common, but on the most chemically tractable residue. And so we began by studying this structure, and we solved the crystal structure of the free protein. Like many other structures that have been solved, it has the nucleotide, it has the switch regions, and here you can see this is the GDP state, the off state of the protein. And switch 2 is shown with a dashed line because this part of the protein was very flexible in this form of nucleotide state. And as you can see, there does not seem to be any nice pocket or crevice where you could bind a small molecule. And we knew this, we expected this from other structures. So we needed an approach to search through a very special chemical library to get a starting point to begin our drug discovery. And so we knew that we were going to make covalent bonds, and so we picked a very particular chemical library that my colleague Jim Wells at UCSF had developed at a company called Synesis Pharmaceuticals a number of years before. And this has a very a special kind of chemical library. And this library is composed of disulfides. And these disulfides have a solubilizing group as well as a drug fragment approach on the members of the library. And then since the protein has a cysteine, you can exchange disulfides. And what we're looking for is a member of the library that has a drug fragment that binds in a pocket adjacent to the cysteine and makes a stable disulfide. Disulfide exchanges happen very easily, so we want to screen and push the system to give us the best binders. And so you can add beta-mercaptoethanol as a competitor, and you can see that beta-mercaptoethanol has a hydroxyl group, so it has no way to fit into the adjacent pocket. So it's a test of a simple disulfide or a special kind of disulfide. And this library had about 500 members, and once we screened those using full protein mass spectrometry, so we know exactly the identity of the molecule attached, we picked out 2 molecules that bound to 100% of the mutant protein, but did not bind at all to the wild type. And there are other cysteines on the protein. Nonetheless, this only picked out the glycine 12 residue. And so this was fantastic. It was a very good starting point. But actually, this form of the protein was in the off state, the GDP state of the protein. And so we turned the protein into the GTP state and asked, oh, these two, hopefully they'll bind, they should bind, they could bind, and they would be, the real target would be the GTP state. But as you see, we almost got no binding. This was a very sort of frustrating day because this told us that maybe this will work in a test tube biochemically, but it won't work in cells because I told you the glycine 12 mutation is really locked in the GTP state because it doesn't have access to the gap-mediated hydrolysis. And so we took this form of the protein, went back to the collection, and rescreened all 500 compounds, assuming we'll find a different starting point. Instead, we found nothing. No molecule from the collection bound to that form of the protein. And so we were very frustrated because all of the textbook pictures really told us that we should have found we need a drug that binds to the GTP state. But what I don't show you in another slide is that actually the G12C turned out to be unusual in that it has somewhat higher intrinsic hydrolysis than other mutants. So it turned out that this was not as much of a liability that we feared, but it kind of taught us that it was good we screened the off state of the protein, slightly different than what we should have done, but sometimes accidents in science are very fortunate. So the real breakthrough was when we solved the crystal structure of it bound to the protein in the GDP state. Here's the G12C, here's the disulfide bond, and amazingly, it's a drug in a pocket. The pocket wasn't there in any previous crystal structure because this pocket is formed by the switch II element folding halfway over the protein. And once it folds over, then it basically makes the shape of the pocket. And this was a real exciting day because it told us that this was a chance. It also told us why this pocket was not accessible in the GTP state, because this form of the protein would close over the pocket. So with that in mind, we began the real hard work of the drug discovery, and we began to sort of explore, how can we make this molecule better? We know that we have a flexible protein, we have a flexible drug, and so we basically realized we had too much flexibility in our small molecule, so the scientists at Araxes began to optimize the molecule, fitting it better and better into the pocket we identified, and rigidifying it until it became the real key lead for the subsequent drug discovery at many companies. And you can see that then once this was really enabled many companies and scientists to go after the target, you can see that many of the next generation molecules had the same piece that would recognize the cysteine to make the covalent bond, and then many of the rigidified elements, but slightly different aspects on the periphery. And these molecules then, these two, went to the clinic. One is called sotorasib and one is called adagrasib. And in the first trials of highly selected patients that had lung cancer and G12C K-Ras mutations, you can see in this so-called waterfall plot where the tumors that grew were on the left, progressively to the complete responding patients on the right. There was about 80% patient benefit and several complete responses. This was fantastic because it's the first direct K-Ras inhibitor. And one thing I didn't stress at the beginning, I forgot, is that because this drug relies on the cysteine mutation to make its attachment to K-Ras, it does not inhibit the wild-type protein in the rest of the body. So it is much more selective than other targeted therapies by virtue of the use of that cysteine. And that was an added benefit, but really it wasn't our prime purpose in exploiting the cysteine at the beginning. So with those responses, we began an effort to see if this was all we could do for K-Ras patients was just focus on cysteine. And we began to think about the other amino acids, and my lab focused on more covalent chemistry. And when you move to the other amino acids like serine, arginine, and aspartate that have covalent possibilities, you go to much weaker nucleophiles, and so the rules and how we do that were really not known by the community. So we had to develop new approaches to go after these amino acids. The serine approach, I'll explain to you, relies on attacking a very small ring system. And when you put rings that are four-membered, you put a lot of strain on the ring. So when something attacks them and they open, they provide a lot of thermodynamic driving force for the reaction. So we relied on strain to go after weaker nucleophiles. And actually, this was inspired by natural molecules. Many of the drugs that we have, either themselves or modifications of them, are taught to us by the evolution of natural products. And these were natural products that have this beta-lactone, and they target threonines or serines in the active sites of these enzymes. Now, these are active site serines, so they're hyperactive nucleophiles. And nonetheless, our serine is not in an active site, so we had to sort of explore whether we could go away from the hyperactivity of nucleophiles. And so we took about 6 years of trial and error until we came upon this molecule, and we reoriented the beta-lactone into this fused system between a 4-membered and a 6-membered ring. And there we saw very good 100% labeling. But if we made any modification to this, like swapping this atom and this atom, and just switching around, the same strain, but slightly different, we saw no labeling. And similarly, when we added substituents that normally you can add and you wouldn't break the reaction, all these were failures. And then we made a 4-membered to 5-membered ring system and got activity back. So what this told us was to go after these less nucleophilic residues, we really had to have precise positioning. So everything was lined up for the attack. And when we solved the crystal structure of the serine covalently bound to that opened up ring system, we could see how all the interactions were correct. And this is only the second K-Ras mutant that ever had a drug attached to it. So this was a very big day for us because it said that it wasn't just cysteine and we could go beyond the cysteine. And we subsequently made improvements. We knew we couldn't change this warhead, so we had to make changes to the pocket, part of the pocket that bound reversibly, and we could then increase the rate of reaction. So you can see this molecule has not too many changes, but the way that it reacts is in 1 or 2 minutes versus an hour. So we could make systematic improvements, and this is common now in the whole field of K-Ras drug discovery. There are a lot of improvements that are possible because of the wealth of chemistry that people have been applying since that early days in the 2010s. And this just shows that we can modulate all the signaling in the expected genotypes. So G12S, we see all inhibition of the downstream signals, but non-G12S, we don't see inhibition. And we checked to see whether we had just made a sort of a firecracker, a reaction that reacts in many ways, or did we make something selective? And this is a list of the proteins, only 4 of them in cells, that react with this molecule, K-Ras G12S, and 2 or 3 enzymes that have active site serines, something we expected. But this is similar to what is in the G12C approved drugs. So with that, I want to move back, actually, to the rest of the pie chart, and I won't have time to tell you about other drugs we've made that target the arginine. This one makes a very nice 5-membered ring, and of course, the one we are extremely excited about is the potential to go after the aspartate. And in the coming months, we'll be able to report that. We're just finalizing that data now. But I want to go back to the patient situation and look at this in a different way. And I highlighted how the complete responses are attractive, but as you can see, many of the patients are really not achieving enough of a benefit. And there are some people that are intrinsically not going to respond. Their tumors have too many different mutations. We're trying to uncover what those mutations are. The whole field is. But there are patients also that have a response, but then because of the genomic instability, they pick up other mutations that make them resistant to the drug. And those reports have just come out in the last couple of years. And this paper by Andy Aguirre's lab, I think, highlights the kind of responses. And in this figure, there are 4 patients diagrammed: patient 1, patient 2, patient 3, patient 4. Patient 1 acquires the kind of mutation we initially expected, and that is a second mutation in the K-Ras that the drug is targeting to prevent the drug from binding. But actually, that's a minority of the kinds of resistance mutations. The most common are exemplified by patients 2 through 4. And in the first column, you see pretreatment. They have almost 100% variable allele frequency of the G12C allele. But after they become resistant, they pick up many mutations in downstream components, but they continue to express the G12C allele. And this is true for this patient and this patient. So what does that mean in the simple diagrams I've been showing you? It means that a patient starts with a K-Ras mutant, but really relatively few other mutations within the RAS pathway. And then the G12C drug binds to the G12C covalently, and it shuts off the signal, diminishing the growth signal that allows the cell to live. But then, through that selective pressure, you pick up other mutations, like a BRAF mutation, that then short circuits the signaling and allows the cell to receive back the signaling. That seems to be the most common genetic way for mutations to pick up. So we began to think about this and think about this from an immune perspective, and we realized since our drug only binds to the cysteine in the tumor cell, it's basically tagging the protein that originally caused the disease, and it continues to be expressed in the tumors. So what can we do that? How can we leverage that? Our idea is to turn this into a so-called neoantigen, or a tumor-specific marker. These are very hard to find. It's very hard to find tumor-specific markers that are unique to tumor cells. I told you how the mutants of the neoantigens are presented, but we realized that this is a very large chemical difference. And if you want the immune system to see non-self, you need to give it a very good signal. And so we wondered, could we use the drug peptide conjugate that comes from K-Ras processing as a neoantigen? So what does that mean? When we zoom into the so-called immunological synapse, we wanted to re-engage T cells. The way we designed to do that is use the class 1 presentation of the peptide, but now our drug is attached to that peptide. So we engineered and developed an antibody that recognizes that complex. We fused it to a CD3 recruiter, or what's called a bispecific T cell engager. It's a synthetic way to regain an immunological synapse and then let T cells kill the cells independent of whether they have other mutations that are driving. So it's a sort of a cross-cutting kind of therapeutic approach. And so there are many steps to this. The first was whether MHC presentation of a drug peptide conjugate. That wasn't known. And we validated in cells that are easy to test what peptides are presented. A true classic peptide for this MHC is presented. The K-Ras peptide is presented. And importantly, the K-Ras with the drug attached is presented. Then we needed an antibody that recognized the complex. We did this with my colleague, Charlie Craig, and that antibody recognizes the drug. Now this is a tool to recruit T cells. And we did that by setting up a mixed population of tumor cells and T cells from humans and ask if the antibody can then recruit that. And we did that in the context of the parent cell, or a cell where we engineered a bypass mechanism that is the kind that comes up in patients. And you can see that we can get killing when there's just the G12C allele, or when we have any other driver mutation as well. So this is a completely new kind of way to think about tumor-specific antigens, and what's nice about it is it's controlled by a small molecule. So if there was any other issue that was giving rise to immune sort of activation, the withdrawal of the drug would completely shut that down. So what I showed you here in the summary is that we have a way to re-engage T cells on this drug-induced tumor-specific antigen. And because not all tumors have T cells replete in the environment, we also, with Mike Evans at UCSF, have used radioligand therapy to bring beta emitters in the proximity of the tumor cell. And now we can have a sort of a field effect of killing tumor cells independent of each individual cell being drug targetable. So we're very excited about this approach, and we think that this will be a potential way to synergize the initial exciting results with the K-Ras G12C inhibitors. So with that, I'll just tell you who did all the work. The two people in the work that started the project and brought it across the finish line were John Ostrom, graduate student, and Ulf Peters, a postdoc, who really I give a lot of credit to for taking on this ambitious project. But once I really explained the importance of K-Ras to them, they just rushed at the problem, which is a true sign of really fantastic scientists. We used that technology I mentioned from my colleague Jim Wells and Michelle Arkin and Adam Renslow. The work on other alleles was pioneered by Xiang Zhang, a postdoc in the lab who did the S and the R alleles. He also worked with Charlie Craig's lab and Charlie's graduate student Peter Rowader on the covalent drug recognition. And Mike Evans and his postdoc Apurva are working right now on the radioligand therapy. And the last thing is, this is just a small part of projects in the lab, and I want to thank the entire lab. This is from a lab reunion we had a number of years ago with the scientists and their families and kids, and it also helps me sort of thank my own family, including my wife Deborah, who's here, and my kids' families, you know, really help and friends support scientists when the day-to-day challenges of failed experiments come up, but it allows us, I think the support we get from the families are really, really important, and we value that a lot. I value that a lot in my lab. And thank you very, very much for the award and for your attention.